Welcome to the channel. Today I'm going to be running a flow cytometer. So that means we get to use the fun multi-channel pipette. We get to play with these 96 well plates. And of course we get to use the flow cytometer. The flow cytometer is a microfluidics and laser device. It's so complicated. It uses a bunch of sort of new cutting edge technology to count cells one by one and sort of analyze them one by one based on their structure or how much light they're producing or their fluorescence or something like this. So this is our flow cytometer right here. It's about the cheapest one you can get that works well. And it's a huge crybaby. It's just, it's a huge baby. It needs to be taken care of really nice or everything will break. And there's some good reasons why it's a huge baby. By the way, it's an optical illusion if you see that I'm wearing shorts in the lab. They're just skin colored, I promise. It's Saturday morning, come on guys. Before we dive too deep into how we're actually gonna use the flow cytometer, I just wanna talk for a second about what it can be used for. So it can be used for things, you know, like in research laboratories to characterize cells for experiments, either with fluorescence or luminescence. So if you, for example, luciferase will make the cells luminesce, and GFP or RFP, these are fluorescent proteins that will make the cells fluoresce. And so that can be important when you're looking at gene expression or protein production or something like that. And in clinical settings, you can use flow cytometry to diagnose disease, in particular cancer. So a cancer cell, when you shine a laser at it, is going to have different characteristics than a healthy cell a lot of times, and different cell types in mammals and you know in humans, of course, are going to have different characteristics. And when you shine a laser light at them, right? So usually there's two metrics that are used: forward scatter and side scatter. So basically, these are looking at how light interacts with each individual cell. And since you can count, you know, thousands of cells in just a second, you can see the clustering of different cell types, and different cell types will have characteristic forward scatter and side scatter. So you can see, you know, maybe this population is white blood cells, this population is red blood cells, this population is cancerous, this population is non-cancerous. So it's a really useful tool for diagnostics as well as for use in research laboratories. This machine is a baby because it uses microfluidics, which is, you know, a relatively new field. It uses lasers, so it has an actual laser table in the machine, and it uses software and a simple CNC machine that was designed by freaking biologists. So, you know, it's not the most reliable thing ever. When I walked into the room, I made sure to turn this on because it takes a full 10 to 15 minutes just to start the thing. The machine has a crazy amount of cleaning stages you have to do. Just when you start it up, it does a lot of cleaning. And then our protocol, we make sure to run additional cleaning steps before we use the machine. So all in all, just to start the machine, it takes a solid 45 minutes to an hour. So we'll start the machine, it'll run some cleaning protocols, and then we'll do a sip clean step, a back flush step, and a run clean fluidics step. And then we do the instrument quality control, or the instrument QC, and that just makes sure that you know, we're getting the results that we expect to see. Another thing you should do when you're running a flow cytometer is to make sure all of the fluids are topped off. So this is the waste container here. This is sheath fluid, which is basically just dilute bleach, I'm pretty sure. There's a detergent solution and a Fax Clean solution is what it's called. All of these solutions, you have to buy the little bottles of the solutions and then mix them with distilled water and then put them in the machine. We'll also need to change the filter. So, you know, the sort of mechanism of a flow cytometer is it's gonna flow, you know, your solution past a detector, and hopefully the fluid flow is isolating individual cells that flow by. And so the detector can see one cell at a time, and it can count something like 10,000 cells in 1 second, which is pretty cool. So today what we need to measure is GFP, or green fluorescent protein. So it's a fluorescence, and the green fluorescence occurs at 510 nanometers. So in here, actually in the machine, there's a 510 nanometer filter, but in order to do the calibration, you need to use a 533 nanometer filter, which is this one right here. That's upside down. This is a 533 nanometer filter. I'll just spray it with compressed air before I install it, but it's still running the startup protocol, so we'll let that finish. And in the meantime, usually this is what I do, is while it's running all of its cleaning stages, I can set up the plate or plates that I need to run, and get the quality control sample ready and all of that. So what we'll need is, usually what you need to do is wash the media off of the cells that you want to test. So the way that we'll do that, usually, the cells that you have are way too dense to run directly in the machine anyway. So what I'll do is get one of these reagent reservoirs, add some water to it. This is sterile water, but probably any distilled water will do. And we're going to add 150 microliters of sterile water to 50 microliters of the cells that we want to measure. So I'll mark this plate 1, and we get to use the fun multi-channel. So again, 150 microliters of the sterile water to 50 microliters of the cells. See? Fun. And we're going to be running a full plate today. Running one plate takes about 45 minutes to an hour, depending on how concentrated your cells are. So not too bad for running 96 samples. I'll put on the screen here, hopefully, if I remember the sort of graph that we're trying to make, but the test that we're running is what we're calling a titration curve. So we want to see the response of these particular biosensors that we're working with to a particular chemical. So the biosensor that we're working with should activate the biosensor should be activated by the chemical that we're adding, and if we add more of the chemical, the biosensor should become more activated, and if there's less of the chemical, it should be less activated. So that's what we're hoping to see today. So we'll check these cells. They look like they grew. Maybe not super well, but they grew. Hopefully you can see that. So they're probably at an OD of, these are probably an OD of 10, maybe these are 15. So going, these columns are the different strains in triplicate, and then the rows are a different concentration of chemical, and these two we don't need. So I'm just going to use 6 pipette tips and go down the rows, and then go down the rows here. So 6. And I'll pipette up and down just to make sure it's mixed, because I pulled this out of the incubator a few minutes ago. Check if the flow cytometer is ready for me. Okay, it's ready for me. Let's go check it. All right, so we did the first 6 wells. And usually this is the chaos of running the flow cytometer. Oh, good. Fluix air may occur upon installation. Okay, wonderful. Like I said, this machine is a baby, and it's saying it has fluidics issues. I see a little bit of air in there, so we'll do a back flush cycle, and hopefully that will fix everything. But yeah, this is the laser table here. These are all the fluidics. These are called peristaltic pumps. They're pretty cool. So these are the lenses. So before I forget, I'll swap that out. Boink, boink. Blow it off. Install that. All of our fluid levels are fine, so I'm not sure why it's freaking out. So, we'll do a back flush cycle. So usually if there's an air bubble, you can just run a back flush cycle or 2 or 3, and then everything will be fine. Let's actually see in there. Yeah, all of the fluids are fine. So there's 3 tubes in there, one with water, one with Fax Clean solution, and another one with water. And you just need to make sure those are topped up to the appropriate levels, or you could get air in the lines. You can also get air in the lines if your fluid level is too low in the wells that you're testing. But I ran it yesterday, and I know all of the wells that I tested had fluid in them because I got data for all of them, so I'm not really sure how air could have gotten in the lines. So anyway, we'll let it run a back flush cycle. Okay, so I ran 3 back flush cycles. We're gonna run a sip clean. We'll see if it gets mad still. We'll let that run. That takes another 10 minutes or so. So we'll get back to preparing these samples. So this plate is done for now. And so we'll take from this plate grew. Very good. So this is just some more details on this experiment. This is selective media. So this is synthetic defined media, or SD media, deficient in uracil. So uracil is just an amino acid, and the plasmid that has the biosensor has a marker on it that allows the cells to produce uracil. So if the cells have the plasmid, they can grow in this media. So basically we want to maintain the plasmid in this media as it grows, and we do that by saying you have to have that plasmid or you will die. So that's why the media is sort of white. It's not really, or it's white or clear. It's not yellow or anything. Rich media is usually yellow tinted. Okay, so it's running the cleaning step. So this is a centrifuge, and we're actually able to centrifuge this whole plate. So again, like I was saying, we need to wash the cells to get the media off of them so it doesn't bung up our flow cytometer. We're going to run it at 5700 RPM, which is about 5700 G in this centrifuge. 3 minutes. Temperature is going to be ambient temperature, and we need to make sure we balance the centrifuge, or at least try to. So I'm just going to take this excess plate and add a little bit of water to it. Should be good. If it's unbalanced, the centrifuge will tell me, so don't worry about it. So this centrifuge, we can actually change the rotors out on it. So this is a different rotor that holds either 500, I think they're 500 ml bottles, or 50 ml tubes with the inserts. And we have another rotor that holds 1.5 ml tubes in case we need to spin at a colder temperature, because this centrifuge does refrigerate. And you can just take out a bolt in the center with this wrench to change out the rotors if you need to. Well, it's not super balanced, but it's running, so it's fine. So the next thing that I want to do is set up the names of all of the cells, so I'll do that right now. Okay, nice transition, huh? So we needed to label all of the wells in the 96 well plate, and you'll see I'll copy that over in a second, but these are all the labels of all the wells. Basically, in the columns, again, we have different strains, and each half of the plate is going to be a different chemical, so we have the chemical name as well. And then we have the chemical concentration. So DMSO is our 0 control, and then we have 0.1 nanomolar, 1 nanomolar, 10 nanomolar, so on and so forth. And each one is in triplicate, so we have 1, 2, 3. I concatenate all the names, so they're all put together nicely here, and then I can copy and paste these into the software when it's ready. The flow cytometer is still running the sip clean step. It's on the second stage of that, so it should be done in a second. And the centrifuge has just finished. It's at speed 0, and now we can open the door. I'll turn off the centrifuge. So maybe you can see there's a tiny pellet at the bottom of each of these wells. It's pretty small in some of them. So we'll take this, and to dump out the excess liquid, that's it. Just flick it out. And there's going to be a tiny bit of liquid still left, maybe like 20 microliters or something, but that's okay. So then we need to resuspend in 200 microliters of water. It's 200 microliters. And I'll try not to touch the tips into the wells so that I don't cross-contaminate. Tell me what you guys think about pipetting robots. I don't think there's that many tasks that it can do unless you're doing like clinical testing or something like that. But if you're in a research setting, I feel like you need to pivot so much and change what you're doing so often that it's easier just to do it yourself. Like, yeah, maybe I need to do this experiment like 20 or 30 times while I'm doing my PhD, but it's easier for me just to do it those 20 or 30 times rather than learning how to program a robot and actually programming the protocol and then giving it all the reagents it needs and things like that. I just don't see how that's faster. Because we have a pipetting robot in the room adjacent to here, and I just don't know what to do with it. And again, I'm going down in these rows because it's increasing chemical concentration, and also, hopefully, if the experiment worked, increasing GFP fluorescence. Okay, so the sip clean stage just finished, and it didn't throw any errors. So, while it's finishing, it's moving to the next task. We'll do a back flush. That takes about 30 seconds. I'm gonna throw away this old plate so I don't get it confused. The flow cytometer is running the clean fluidic cycle. I already ran the back flush before that. So now I'm going to be preparing the quality control sample. So we'll take one of these 1.5 mil microcentrifuge tubes, and we'll use 500 microliters of sterile water with 2 drops, I know, very scientific, 2 drops of the calibration solution. And the calibration solution is basically just these microscopic beads. I'll show you here. They need to be stored in the refrigerator. So these are beads. And we'll take one of these. I'll keep the other one in the fridge. We'll take one of these bottles, and you know, there's a good chance that they're clumped at the bottom or something like that, or stuck to the walls of the tube or something. So we'll vortex the hell out of them, shake them up real good. Oops. Vortex it really, really good. And then we'll take this and we'll put a couple of drops. So 1, 2. I have no clue why they can't just give you a tube and you pipette out the appropriate amount. I don't know if they don't trust you, or maybe the cells very quickly fall to the bottom of the tube and so you need to hold it upside down to get the beads out properly. But either way, that's our calibration solution. We'll vortex and shake this up too. And then this will be ready to go. Again, we need to run our quality control at with the 533 nanometer filter, so that's already in the machine. And then once the QC is done, we'll switch it to the 510 nanometer filter so that we can measure GFP. So we'll put this in the A1 slot of this little, what is this, 6x4, 24 well plate. All right, so the flow cytometer is ready. We'll eject the plate and open the instrument QC panel, and we'll put this 24 well plate in. Again, we're checking that these fluid levels are at least to the 2 milliliter line. This one is really high, but it's fine. This one is also very full. It's supposed to be, this one actually fills up. It's a waste and back flush tube, so we'll just dump off a little bit of that. That plate I need to throw away, and we'll do run and proceed. So we'll throw this plate away. So it's going to be first measuring the noise, so just how many particles it sees flowing by just in the wash solution. So you can see there is the noise measurement, and we expect 7,500 events within 20 seconds. And if we don't get to that, then there's some issues. So you see it's counting, there's the noise on all of the graphs, pretty cool. And once it gets to 7,500 counts or 20 seconds, then it will start counting the beads. And within 5 minutes, it needs to collect 25,000 events. If it doesn't collect those 25,000 events, then there's some issue with the noise measurement or the beads that were prepared, something like that. So it expects a certain amount of beads in this dim region and then this mid region. And you can see the axes here are side scatter, so SSC, and forward scatter, just like I was talking about towards the beginning of the video. And then it's also measuring on the 4 different detectors. So there's FL1, which is where we put our 533 nanometer filter, and we'll switch it to the 510 in a minute. But there's also 3 other filters in here, so FL2, FL3, FL4. We can scroll down, and once we get the report, it will, add up all of the counts that it got in those ranges and say if it's within the range. So our machine pretty much always, and you can see this is live, it's counting them right now and adding to the regions. But our machine always overcounts a little bit on the forward scatter of the beads in this range in particular. And I'm not sure why. It just does. Maybe I need to change this box. But it overcounts just by a little bit. Like the acceptable range is 411,000 to 764,000, and it usually counts like 790,000. So it's pretty close most of the time, and it's pretty rare that it passes. So anyway, it's up to 10,000 events now, and it's been a minute and a half, so we're gonna get to the 25,000 events within 5 minutes, so that will be great. And actually, just to prepare, I'm gonna copy the names of all the wells so that we're ready when we run our plate. Another important thing to take note of here is that these are V bottom plates. So the bottom of them, hopefully you can see, has a little V. The bottom of the plate is like this. It's a V-shaped well. And that's going to be really important because we need to tell the machine that we're running with V bottom plates so that the needle knows where to go or how deep to go into into the well. Okay, so it's almost done. 24,009, 25,000. Wow, we passed. Okay, it's because the camera's on. So 759,000 events, and the max was 764,000 events. So it passed the test. It's doing a little rinse right now, and then we'll be ready to set up our plate. Cool, that's awesome, it passed. Okay, so we can close this, we can confirm the regions. And then we'll close that. We want to go to auto collect. Again, we want to select this V bottom plate. I'll save it in my folder and name it appropriately. All right, so we need to select all the wells we're going to use. We'll do select all since we're using all the wells. We'll run the fluidics fast. And these are the run limits. So we want to count 10,000 events within 1 minute and within 100 microliters because we only have 200 microliters in there. We can do, let's see, we won't do that. We'll do apply settings. So it's with the run limits, it's going to stop whenever it hits one of those limits. So if it hits 10,000 events, or it's measured for 1 minute, or it's measured 100 microliters of volume, it will stop. So we'll do open run display. Actually, we need to eject the plate first and put in the appropriate plate. And I almost forgot, we need to put in the appropriate filter. So bye-bye 533, hello 510. GFP fluorescence is at 510 nanometers. So we'll put that in. Take that out. Put this in. And we'll auto run that. So that's it. It's not a particularly difficult machine to use, but if you mess it up, you can mess it up really good. Again, there's a lot of sensitive components in here, and you want to make sure that you set it up properly. So this is something you don't want to use unless you're certain of how you're using it. And one component that you probably saw moving is this sort of CNC component. Usually CNC computer numerical control machines will have an X and a Y axis and then a Z axis. This one has a normal linear Z axis, but it has two rotational axes to get you the X and Y positioning. And it's so like flimsy and floppy. I don't know why it was designed like that, but whatever, it works. And so you'll actually be able to watch each sample here. You see you have side scatter, sit down. So you have side scatter and forward scatter like I was discussing earlier. It's about to start running well 2 already. It's already run well 1. And so we'll see the events rise. There's not many cells in this well for whatever reason, but we're at 3,000 events and you can see it's populating on the graph. 6,000 events within 10 seconds. We've used 15 microliters. Not sure why that well didn't grow very well, but we got to 10,000 events in 20 seconds using about 20 microliters of fluid. Usually I get to 10,000 events in 1 second, but I guess that well didn't grow very well. We're growing these at 42 degrees Celsius, which is a pretty high temperature for this yeast, and they're also in a stressful condition. So that's why there's less cells in the well. Anyway, that's flow cytometry. I'll put my data on the screen here. If you have any questions, leave them in the comments below. Don't forget to subscribe, like the video, all of that. There's plenty more videos like this. I've done several lab POV videos and I plan to do more. If you're interested in a particular topic, let me know down below. Thanks for watching. Bye.